Today, in this tutorial, I will present you how to use sm-dtw, which stands for Split Merge Dynamical Time Warping, a tool that realigns two sequences of plant architecture. So this tool was developed during the ROMI project, the project Robotics for Microfarms, a project which was funded by the Horizon 2020 program of the European Union, and which includes all the partners that you see in this first introductory slide. So in this tutorial, I will first present you what is sm-dtw to know what it is about, and especially I will try to explain the context so as to know why and when you could use and need sm-dtw. And then it will be more practical, so I will explain how to install and more precisely how to not install it, because it's very easy thanks to a Docker image. And then once it's on your computer, we will have a practical session using Jupyter notebooks to use our tools. So first, what is sm-dtw? It stands for Split Merge Dynamical Time Warping. It implements a generic dynamical time warping algorithm to align two sequences together. The use of dynamical time warping algorithms is now widespread. So for example, it's very known for speech recognition when one sentence is said and spoken by two different speakers with different voices, rhythm, etc. And in biology, it has a great success too with the use of BLAST, matching two sequences together, be it like proteins or nucleotides, which can have like differences between them. So the idea is to wrap one sequence onto another, a test sequence, which has diverged from a reference sequence, and to find the place where those two sequences diverges. So generally, two different constraints or different constraints can be applied to edit distance to wrap one sequence onto another. So this can be classical, symmetric, asymmetric. And here, in our new tool, so we implement two new features to adapt this tool to tree phenotyping. So first, a new constraint. Now edit distances must be either splitted or merged, corresponding to either new or lost branching point in an existing tree, which correspond to a best aggregation mapping problem. And a new data type. So in tree phenotyping, we have distances, but also sometimes we have angles. So now sequences can be composed of scalar and angles, which are cyclic data. Okay, so this was a bit generic. So now I will try to explain why and when do you need sm-dtw. So this is a very useful tool for people doing automatic phenotyping of tree structures. So to be concrete, I will take an example. One tree structures are phytotaxis. Phytotaxis are the arrangement of branching structures around the stem. So here you see two beautiful examples of phytotaxis, which are like very well-known geometrical shape that are built by plants. And numerous plant scientists are interested in understanding how such patterns are built. And to characterize those structures, you can measure them, and you can characterize them quite well by two features: either the divergence angle, which is the angles between two successive here leaves, for example, that have been produced, and also internal length, which is the distance between, for example, two leaves here on the stem. Okay. And here I will not propose you to measure phytotaxis on those two beautiful plants. I will propose you to measure phytotaxis on this plant that you see on the left hand side here, which is one of the major plant model plant for plant science, which is Arabidopsis thaliana. And here the idea is that you see the main stem of an inflorescence. The little branching structures here are the fruits, and the idea is to identify the stem, identify the different fruits, and measure for each pair of successive fruits the divergence angle between them and the internal length. Okay, and during the ROMI project, so we developed tools to both image the plant, make all this recognition automatic, and here you see the same plant where the structure of the plant has been recognized and the fruits have been spotted by our software, so they are now highlighted here with these two alternated shades of pink. And then from this you can derive a measure of the angles and the internodes. So quite easy. But now when you're a biologist, you have a question. So you want to check whether the computer that gave you this result is true or not, whether these sequences are the real measures that you have here. So to check this, you can use like a manual measurement, so a second measurement. So for example, here we built a device with a protractor, and with the protractor, for example, here you can measure the angles between two successive angles. Using a ruler, you can also measure the internal length, and you end up with manual measures. And if you are quite self-confident about yourself and your measures, you can say that this is a grand truth. So you trust this kind of manual measures. And you can compare the manual measures with the automated measures. And at the first sight here, you can see that despite the fact that the segmentation on the left-hand side were quite well, so all the fruits were quite well recognized, you see that you have differences between the two sequences. So this is not really cool. And when, so here in this plot, I realign the two sequences together, starting at the same point, and you can see that clearly they are not the same, they diverge. So, which is a bit annoying. But now when you look a bit more closer, you can see that, for example, so when you see this pattern, you find it here again, for example. Or when you see this pattern here, you can see here. So it seems that some sequences have been just shifted and some have been added. So analyzing this type of two sequences and trying to match, to wrap the automated sequence onto the manual can be quite complicated, tedious, and time consuming. So that's why, instead of doing it by hand, the idea is just to use sm-dtw. So to sum up, why and when do I need sm-dtw? So to use it, you need two types of input data. So you need two input data, two measures of the same branching tree. For example, a grand truth versus a test measures. And the expected output will be that it will automatically detect the differences in the branching point, their position, type, and compute the differences in the measure position. So for example, here you will realize that the differences that you see in your sequences are because each time on the left hand side I spotted some stars here, you have like segmentation errors. So fruits that have been recognized and should not, or fruits that have been missed. And this causes, of course, perturbation in the sequence. And so it would be very easy if the program could automatically detect on the stem those errors. So in what kind of possible scenarios people would require such a tool? So for example, if you are benchmarking two analysis methods, or you're trying to assess the result of your new method, or you're trying to improve it and develop new features in your method. So the target user are, of course, biologists, computer vision scientists that are either testing and or developing tools for automatic tree phenotyping. Just before starting the more practical session, I recommend you to go to the GitHub repository of this project, because you could find a written README that will be very helpful as a complementary help to this video tutorial, and especially just to copy and paste some a few command lines that we may need to use during this tutorial. So the first part is to install our tools to us to work with it. And it's very easy because in fact you don't need to install it, you just need to use Docker. So, and to use Docker, you need to install Docker in your computer, and I just give you the link. It's very easy to install. Basically, it's a few command lines depending on your operating system, and I give you the link to the very, very beautiful and pedagogic documentation of Docker to install Docker on your computer. Once you have Docker in your computer, then you can download our Docker image, which is on a hub, on the cloud, available for anybody freely, and it is you will have to type this very simple command line, which is docker pull robotics microfarm sm-dtw demo with the tag latest. And you can also try to run the Docker directly, and if you have not pulled already this Docker image in your local machine, it will download it. It will check the repository in Docker Hub and download it directly into the local machine. And so now let's start the real practical session, hands-on. So as I told you, maybe the best is to open a web browser and to go to the GitHub page of this project, which is hosted on my account, and then you go to Docker subfolder and you open the README. In the README, you go to section 2, and this section 2 first asks you to create a folder where you will store all the data created by this tutorial. So you open a terminal. Here, for example, I will change to my desktop, and as it is mentioned here, I will create here a new folder, okay, and I change in this folder, okay. And so now you can start the container, and to start the container, you just copy paste. Oh, sorry. You just copy paste this command line, and you will see that directly the container will start, because the prompt has changed. Okay, so now you're in the container, and this DTW in brackets means that all the environment which is required to use our program has been loaded. And then this is the last command line you will have to type. You type the second command line here to start Jupyter Notebook. Okay, you click, and then you will see different things printed here, and especially like here an URL starting with 1270. So you control click on this URL, and this will automatically open the page in your web browser, and you now see like you have different notebooks, and we will go through all these 3 notebooks here: step 1, step 2, and step 3. Okay, so let's start step 1. So in this first part, you remember that sm-dtw, as input data, you need 2 sequences, one reference sequence and one test sequence. So here we will generate these sequences, Phytotaxis data, and we will make like synthetic Phytotaxis data. So before starting, just be sure here that your kernel is in R. So you go to kernel here, and you see that you can select different language for your kernel: Bash, Python, and R. So here it should be R, and in fact when it's R here, you can see that the R logo and or R here is mentioned on the top right hand side of your Jupyter Notebook. So in this tutorial you will see that you have like different cells containing either text or code. So the text is quite self-explanatory, so you can read. You have like basically a written version of what I will saying. So I will go quite fast on the text cells to concentrate on how you can modify the code cells. And so basically, to run a Jupyter Notebook, you just need to click successively on the run sequence here. Okay, that will drive you successively through all the different cells. So here, let's go to this cell, because here that's where all it starts. Here we will generate one Phytotaxis sequence, which will be our kind of reference sequence. So here you just have to define the length of your sequence. For example, 20 intervals means that a sequence is made of so 19 intervals means that a sequence is made of 20 organs. You can choose the divergence angle, the mean divergence angle, the variation of this of natural angles around this mean, and you can also choose the noise on the internode length that will shape the distribution. Yeah, okay, and all this will be incorporated in a sequence here, or in a function that will create your reference sequence. So you can play around with those different parameters. You have like extra parameters that are indicated here, okay, and that allows you to modify a bit the sequence. The default values that we implemented have been chosen so that the final sequence looks like a typical Arabidopsis Phytotaxis sequences. And there may be one trait which is not self-explanatory here, which is called natural permutation. So I will just explain what natural permutations here are. So in fact, naturally, it happens that the order, so this is a stem, those are the little fruits of Arabidopsis, and it happens sometimes that the order, the standard order of organs along the stem is changed in a stem, and instead, for example, of this number 2 being below the number 3, it happens to be above. And then if each organ is separated by an angle of 137 degree, okay, so this permutation here in the order along the stem will create a typical M-shaped motif, okay. And for example, here I plot you a real measurement of a real plant, Arabidopsis, and you can see that indeed you can see typical M-shaped motif in the sequences of plant. And you can even find like plants, genetic backgrounds, where the number of such permutations is increased, okay. So here we set up a frequency of natural permutation which is close to what you see in Arabidopsis, and if you increase this permutation, you will see more and more like kind of accident and M-shaped motif in your sequences. Okay, so once you're happy with the basic parameter of your sequence, so I advise you here to start just by playing with the length of the sequence. Maybe you can start the sequence, so you have information telling you that the sequence has been generated, and you can plot it just to observe it. Okay, so here you have the sequence of angles and internodes. Okay, so you remember that to use sm-dtw we need like 2 sequences, so one reference and one test sequence which has been modified. And here you have like 3 sources of modification. So either segmentation errors, so that are basically fruits here that will be either missed or added. Permutations here. Permutations are like natural permutation, inversion in the order of organ, but not natural. This one, it will be induced by your measurement. And noise, this is just measurement noise here. Okay? And to compute these 3 types, these 3 origins for errors in the test sequence, you will have to choose your scenario. So you can choose either segmentation errors set to true or false, permutations set to true or false, and noise or measures set to either noise or measures. So for this particular point, I will just explain what I mean by these 2 scenarios. Okay, so these 2 scenarios have been implemented to kind of mimic a real phenotyping scenario. So, and it concerns mostly the difference in which the measure values is implemented. So let's look at the first scenario, noise. So just imagine you've got your plant, and from your plant you measure with the first method, like let's say manually, you measure angles and internodes. Okay. The noise scenario will say that it will implement noise here, a tunable random Gaussian noise on angles values and internode values. And you will generate the test sequence, and the test sequence here, if you consider that you have no segmentation error, no permutations added, so only noise, this noise will come from directly noise applied to the values of this reference sequence. Then, what is measures? So now it's more what you have in reality. So you have a plant, and this plant has its real and own divergence angles and internode sequence. Okay, but in fact you don't know what are those values. What you can do only is to try your favorite measurement method and to measure it. And these measurement methods has also some noise, imprecision, etc. Okay, so let's say it's our golden standard or manual measure here, for example. And then you develop a new tool, which is the measure 2. And from the same inaccessible real plant, you derive a second measure. Okay, and here the both reference and test are derived from this inaccessible real sequence. But here, what you can do in the program is to tune separately the noise of measure 1 and the noise of measure 2. Basically, it will be like you test that your golden standard has 1 precision and your new measure method that you want to assess as another precision. Okay, so let's go back to the let's go back to our tutorial. Step 1. So for example, here I suggest that we use these measures, for example, and so we make segmentation errors. Okay, so that's the scenario. Okay, and now we will choose which kind of segmentation error we will implement. So it is here, and to explain this I will again turn out to a small presentation to explain how you can encode segmentation errors. So let's take a plant like this, and I numbered the different calyx, the different fruits here by the number. So that's the fruit number 6, 7, 8, 9. Okay, so what you're trying to do again is to measure the distance between two successive fruits here and the divergence angle. Okay, so now imagine that your program is trying to automatically recognize stem and fruits, but it makes some errors. Okay, what kind of errors it makes? Sometimes it adds artifact, so an organ which is not an organ, and sometimes it is missing fruits. So imagine it's adding an organ here between the 6 and the 7. It is missing the 7. It is adding 2 organs in between the 8 and the 9th calyx. It is missing the 11, and it is adding a new organ after the 13, for example, and the rest of the sequence isn't changed. So how can you encode these modifications? That's what you want to implement. Okay. So in the previous tutorial notebook, what you will do is to, for gain and losses, indicate the numbers you want to change. And there are some subtleties. So in gain, if you want to add a new fruit here between the 6 and the 7, you will tell it by putting a number here, which will be the number that this fruit will have in the new sequence. So for example, if I put it just after the 6, it will be the number 7. Okay, but of course you don't need to recompute and to propagate all the changes. So for example, here I put number 7, and then I want to introduce 2 new organs between the 8 and the 9. So I say I want to introduce just after the 8, so it will be the number 9. So you put number 9. And if you want to put another one, like 2 in between the number 9, so you put again 9, and as much as you want here, and all the new calyx will be like packed in the small interval here between the 8 and the 9. And then again, if you want to add an organ after the 13th fruits here, it will be encoded as 14. For loss, it's a bit easier. You just type the name in the reference sequence, the number in the reference sequence of the calyx you want to remove. Okay? So if you apply those changes, that's what you will have. Like the removed calyx will be removed, and the new added calyx will be added somewhere in between where you said it. And so when it's added like just after the 7, so like number 7 here, it will be added like in this small interval. Okay, and so necessarily like the internal length should be comprised should be shorter than the previous internal length. Okay, it will be in this interval. But the divergence angle is random, so it can be like randomly inserted anywhere on the horizontal plane. Okay? So for example, here we will implement gain and loss sum function. So for example, it's very often that you have like errors like this. For example, so here I have a sequence of 19 intervals. So you can say that you lose the 3 last intervals, 3 last organs, okay, and also the 2 first one. And in the middle, you add some organs, okay? Why not? So in our scenario, we don't have like any induced permutations, and we have measures, okay? And here we can set up like the precision measure of the first method is the precision measure of the second method, okay? And this tells you whether this figure are encoded into a percent basically of the standard deviation mean, or are just absolute values. Okay, so here we define the errors that we introduce in the test sequence. and then we will generate the sequences. So here you have like printed message, okay, and you can plot the sequences. So when you plot the sequences here, you can choose to plot either the sec reference or even like also the test value. So you can see here that after adding those issues, like those segmentation errors and the different divergence in terms of measures, the two sequences are diverging a bit. Okay. So now you can also try to plot them like starting together on the same plot. Okay, so the differences are more obvious. So the second measurement, the test sequence, is in blue, and the first measurement is in red here. Okay. And since you know that the type of errors you added, you can realign the sequences based on the error you implemented. And so here, for example, so I will just zoom a bit. You can see that you have, like, for example, here tilde sequence, meaning that those two intervals matches. Okay, so the difference between them is only due to differences in measures. You can see also that at the beginning of the sequence, so you've got organs that have been missed. So in the measurement, so you remember that we removed some organ at the beginning, and you can see M. So removed organ are seen either as chops, C, when they are at the tails of the sequences, or split when they are inside the sequences. And you remember also that we added some organs, okay, and when we added those organs, this creates merges here, little M's on a vertical line. Okay, so all this is explained here in the comments below the multisec plot. Okay. If you want to understand also to check what happens at the organ level, okay, so here you can see a table that explain you, like here, a number with the reference organ and the modified organ. And you can see here, for example, that this number 15 here, okay, organ, in the test sequence, in the modified sequence, was added. It has no counterpart in the reference sequence. Okay, so you can view this table at the level of the organs, but also at the level of the intervals. Okay. And finally, what you will do is to export your result in the Docker sandbox folder that you just made. Okay. And at the end of this step 1 tutorial, now we have synthetic data, one reference, one test sequence that diverge because of errors. So after this step 1, we will start now the step 2, which is the second Jupyter notebook. So first you can check, then opening your file system, that in the folder that you created, Docker Sandbox, the right files have been created, and especially the reference sequence and the test sequence that contains the modification that we implemented, like a gain and loss of organs, okay, and the two tables that align the organs or the intervals. Okay, so let's go back to the home page of the Jupyter notebook, still serving in the Docker. And so after step 1, we then now open step 2. And here in step 2, we will use DTW to realign the sequences automatically. So again, here I would like you to pay attention to the kernel. So it should be here the Python login and DTW. And if it's not the case, you go to kernel, you change the kernel, and you select DTW. Okay, once it's done, then you can go throughout the different cells thanks to the run button. And so the first step is just like uploading the libraries and uploading also the sequences. Okay, and here you can check that the sequences, either the reference or the test, have been well generated before. Okay, so you will here convert the format, so DTW can use it. And here you will really use now DTW. So first, the first particularity of DTW is that one sequence is made of two vectors, one for divergence angle and one for internal length. And those two values are not of the same time because divergence angles are angles, circular data. And so you have to say that my data will be of mixed type, and true means that it will be angles, and false would mean that it will be like scalar values. Okay? And so basically you don't have to change the other parameters here, and the only thing that you can change here is the weight that you give to divergence angles and internal length. So you can play with it, saying that you will give more importance to the divergence angle or to the internal length to wrap the two sequences, okay, in the combinatorial model. So here, to keep it simple, we give exactly the same weight to the two vectors in the sequence. Okay, and now, so you run the cell. So each time a cell is run, it should a number should appear here, meaning that it runs successfully. And then everything is set up so you can run the first realignment. So here we run the realignment, and we will see the results here. So you print the results. So it gives you information like the test of the length sequence, of the reference sequence. It reminds you that you use a merge split constraint, okay? And here you have like the result of the alignment, okay? You can see it in a table, okay? But maybe the easiest thing is to use a plot. And here you see that the reference sequence is in blue, the test sequence is in orange here, and you can see that it recognizes like matching points, okay, and also like split and merge events. Split are arriving because an organ has been missed in the test sequence, and merge because an organ has been added, okay? And this is true for angles and internal sequence. You remember that in the sequence we just previously generated, we missed some organ at the beginning and at the end. And this is very crucial when we are trying to wrap those two sequences onto each other, to be explicit about your bounding conditions. And here, the bounding conditions are set up so that the two first points and the two end points should merge, or at least should be kind of aligned. But you can say that, okay, if I missed too much organ at the end or at the beginning, I can remove these organs from the analysis and try to align only the internal part of the sequence. And in our case, it's very crucial because beginning and end of the sequences are more error prone than the central part of the segmentation. It's because that's at the border of images, also because you have like young organs which are very tiny at the tip of plants, for example. So there are many reasons why segmentation should be more problematic at the beginning or at the end, and it's very important to define precisely free ends. So here you have like conditions for free ends, but what you can do first is to play with free ends. So you can check the current value. So the current value 0, 1 means that basically you have no free ends. And you can change the free ends. For example, you replace this with 5 and 5. So here you can, in this cell, read the conditions on the values that are allowed for free ends and their meaning. But basically here you increase the free ends possibly. So basically you can drop the end of the reference or test sequence and try to only align the central part here. And you can see that when you do this, compared to this plot has changed, and basically you have less sequence in the test sequence that are aligned now because some of the sequence has been dropped. Okay? The problem with this is that you have like a certain number of free ends at the beginning and at the end that you could use, that you could test, and it's very time consuming to test all possible free end values. So in DTW, our program, we implemented a program that automatically found and select the best or the optimum free end conditions. So here there's a text to explain you how this works, and basically here that's the code. You have two main parameters like freehands epsilon and freehands number here. So the freehands epsilon here, it gives you a number, and this number is the sensitivity. So you accept a freehand, so you accept to chop your bounds of the sequences only if the next solution is better than the previous one, based on the normalized cost of the alignment. And to determine whether the next solution is better than the previous one, they should not exceed a certain value, and this value is determined by freehand steps parameter. So the lower it is, the easier it is to increase freehands basically. And here the freehands parameters, so you saw before that this parameter can be a tuple, like to indicate the freehand at the beginning and at the end of the sequence, but it can also be a float. And what is a float? It means that, okay, you can chop ends at both ends of sequences, but not like you cannot chop the entire sequence. So this is to avoid that minimal sequences that would be perfectly aligned but very meaningless. So to avoid this, we say that the freehands search will be capped to a certain value. By default, it is capped to 40% of the length of the shortest sequence, and here you can reduce this even, like if you don't want to have like extremely high freehands. So for example, here the search will be capped and ended once we reach 30% of the shortest sequence. Okay, so you can modify this sensitivity and the range of freehand search. You run it. So this takes a bit more time than the previous one. When it will be done, so the star will be stopped here, and also the DTW kernel running will become white again. So now it's done. A number appears in the cell, and you've got the result here. So automatic selection found that the best, according to our program, the best freehand combination was 1,7. Okay, with this normalized cost. You can then rerun DTW with this freehands combination. So the 1,7 here has been stored here, and now that's the new plot that you observe. So the best combination is to have like a bit of freehand at the beginning and also at the end. Okay, so this is the best alignment DTW proposes you, and this alignment is aware to the structure we have, like it is aware of angles, and it is aware of segmentation errors, because the way it is wrapping one sequence into another is by incorporating this constraint, okay, the split merge constraint, that if you add an organ, it must be necessarily between the two previous organs, and if you lose an organ, the new internode will be the sum of the two previous one. Okay. So once you have this, we'll be able in the next part to check that the alignment of DTW has been correctly done. So you export your results. You can verify that the table that you exported is correctly done. You can verify that here a new table has been created, and we will now compare the alignment proposed by sm-dtw by the grand truth alignment that you generated in step 1. So now let's go to the third part of this tutorial, step 3, Jupyter Notebook. So you go back to the main page of the Jupyter Notebooks. Now you open step 3. This time this kernel is running with R, so verify that R is the selected language for the kernel, or else you can change it here. So now what we will do is that we will compare the alignment made by sm-dtw with the alignment we programmed in the synthetic data we generated at the beginning in step 1. So we have to upload all this data together in this Jupyter notebook. So that's what we do here. We import data simulation from step 1 and alignment prediction from step 2. Okay. And so what the program does is that it converts a bit the different format of the output of this sm-dtw with the format we used for interpreting phyllotaxis sequence in step 1, so that we can understand each other. Okay. So all these steps is this, basically importing the data and converting them to the right format. Now step 3, we will evaluate the alignment prediction made by sm-dtw. So by running this function, okay, you can see that very quickly you've got a printed results, a table, and here for each interval we will compare the prediction made by sm-dtw with our simulation. You can look at it on the table, so you see there is one error, one mistake at the beginning, but then everything is correct, and then you can like look at it at the table, but it's more practical to look at it on a plot. So here with this function in the section 3.2 you can generate a plot. So don't pay attention to the warning messages, just explain you that you might have freehands somewhere. And here you have, like, on the top you have angles and internodes that you simulated with the correct labeling of merges, split, chops, okay, tails. Okay, that's what you created. And below here you've got the prediction which is made by sm-dtw. And you can see that in this prediction some parts are overlaid in ray and some parts are left in blank. So when you have blank parts, it means that you have a perfect match with what you implemented, what you simulated. So sm-dtw find exactly the right matching part. Okay. And when you have red parts, you should pay attention to it because maybe you have some problems. So for example, here you had like an organ in this that was so the true organ was removed, but another one was created instead. So very close because you have short internodes, and of course sm-dtw is not able to find these events because there's no way to find such a substitution between a true and a false organ. So it tells that this organ is true, for example. Okay, is correctly matched. So here it's an inambiguity which is impossible to resolve. So sm-dtw makes the right job here, in fact. And here at the end you can see that we had like chops at the end and on the reference sequence, but in fact here sm-dtw prefers to classify this as freehands, and you see that you have like a series of chops and tails, and for DTW, like this pattern is more likely to be completely random than a suite of merges and split. So it does not recognize the correct pattern, which was like one merge, one split, and one chop, two split at the end. But at least it recognizes that the end of the sequence is not matching, and that you have like other organs that have been added that does not correspond to the initial sequence. And so sm-dtw here is a bit restrictive. It's also because maybe this behavior could be like adjusted with the freehands epsilon parameters that we choose with the sensitivity. So here we give kind of a priority to freehands compared to trying to match. But anyway, so the program could find that you have a central core here which is correctly aligned, and it could like precisely identify from which organ, which interval to which interval you've got a perfect segmentation. Okay. So you can summarize this comparison and this performance of DTW in a table. Okay. So you will see here, for example, the number of reference organ, the number of test organ. So you're trying to match 18 organ with 19, so you know that you will be like at least one under segmentation. Okay, you have like the performance of DTW that can align 13 organs, so 13 intervals, like here, out of the entire sequences. And so here you have like the number of intervals that have been matched, the number of intervals or the ratio of intervals that have been aligned but with split and merge operations. And here you have like the number of the longest segment which is correctly labeled by sm-dtw. Okay. And so as we see here, we are quite restrictive. So because even when the alignment is quite correct, here with this comparison, we flagged it as a potentially problematic. Okay. But the cool thing also is that now, so at least we can easily identify where you have like segmentation errors. But if you take only the matching intervals here, you can also kind of evaluate the precision of the measure, like what is the distance between the value of divergence angle or eternal length between the blue sequence and the red sequence. Okay. So by doing this, you can summarize here, for example, in a table that will give you the number of organs that have been missed at the beginning, at the end, the number of over segmentation at the beginning or at the end, based on sm-dtw program. And also at the end here, you will have like the precision of angles and internodes in terms of absolute value or standard deviation. And you can see here that you've got like a quite precise, like the standard deviation is 10 degree here, or 7 millimeter, which is a bit larger. But at least here, the difference between the two values, you got an estimation of this precision. And so if you are not satisfied with the precision, for example, you say for angles it's quite okay, but for internode length it's still quite big, then you can specifically work on increasing the precision of your measure 2 concerning internode length, for example. Okay? And with this, it's at the end of step 3. If you go back to Finder window, you will see that new data have been generated, especially the plot of the comparison, so you can keep track of your sm-dtw. Okay. And with this, so you can loop again to all the 3 steps, so you can close here this notebook, and you can rerun again all the different steps by generating new sequences, aligning them again with DTW, and checking the results, and to test different parameters, either when on the type of data you simulate, and or the parameters of sm-dtw. Okay. Just one comment: when you rerun a kernel, so it will keep in memory your previous run. So I advise you to restart from scratch and to clear the output. So you click kernel, restart, and clear output, and you will see here that all the results of the cells has been erased, and now the cell is turned into non-run yet. Okay. And maybe to finish this this tutorial, so I will go back to the true example, because before, so here in this tutorial we generated synthetic data, so it's not real phototactic sequences, so we generate the sequence we want, but we can use, of course, DTW, our program, to help us to analyze a real phenotyping experiment. So in the previous plant I showed you, You remember that before realignment, I had like clear differences between the manual and automated measures. And using sm-dtw now, we can clearly find segments of perfectly aligned, here in green, measures. We have like segments of over segmentation and segments of under segmentation. And so here you can clearly, on the plant, I highlighted the corresponding parts in the plant where, at least in those two green boxes, you know that the segmentation works perfectly well. And then you know that, like for example, at the base of the plant here, you've got segmentation issue with over segmentations. Tails are like over segmentation added to the beginning of the sequence. You know that you have also over segmentation at the end, and also under segmentation, like some of the real fruits have been missed at the end. Okay, so here you can automatize the analysis of your results in terms of segmentation and precision, because here are the same. You can compute for the correctly matched part what was the precision of your automated measurement in terms of angle measurement or internal length compared to your manual or standard grand truth reference method of measure. So maybe just before finishing this tutorial, I will explain you how to quit everything and to close properly the files. So what you first do in your Jupyter notebook, running, serving on your web browser, you just quit here. So this will stop the Jupyter server. The server is stopped. So now you can just close this browser and you come back to your terminal, and you can see that now Jupyter Notebook has stopped, okay, and you have the prompt of your Docker, which is still running, okay. So now what you can do is close the Docker just by typing exit, and now you see that you close the Docker and you're back to your computer with your normal prompt, okay. And here you remember that you are in your data folder that was mounted in the Docker, so that everything that you produced as data during this Jupyter Notebook session has been stored in your computer, and so you have full access to it afterwards.